Germline mutations in tumor suppressor p53 predispose individuals to a spectrum of cancers, and most frequently sarcomas, gliomas, adrenocortical carcinomas, and others. And this hereditary cancer syndrome is termed Li-Fraumeni syndrome. Of the p53 mutations associated with Li-Fraumeni syndrome, most well studied are those that occur in the DNA binding domain of p53. However, much less well known are how mutations in the oligomerization of domain of p53 contribute to tumorigenesis. We were specifically interested in the A347D mutation, which causes p53 to be unable to form the active form, the tetramer. And instead, this protein preferentially forms dimers and is the only dimer-forming variant among Li-Fraumeni syndrome-associated oligomerization domain mutants. Ultimately, we wanted to understand from a basic science standpoint how the gain of function or loss of function activities of this mutant p53 contributes to tumorigenesis. In order to study this mutation, we used two experimental models. First, we obtained fibroblasts from a Li-Fraumeni syndrome family, and two siblings were heterozygous for the A347D mutation, and one harbored wild-type p53. And our collaborators in the lab of Dongfang Li at UT Health subjected these fibroblasts to the Yamanaka protocol to generate iPSCs. These iPSCs were then differentiated into osteoblasts, which are the cell of origin for osteosarcomas, which are commonly observed in Li-Fraumeni syndrome patients. In addition, we used CRISPR-Cas9 to knock out p53 from U2OS osteosarcoma cells, which are wild type for p53, and generate cells either heterozygous or homozygous for the A347 mutation, or, as I mentioned, we knocked out p53 from these cells. So this gives us an isogenic allelic series to more cleanly study the effects of p53 A347D. And we used more global unbiased approaches such as RNA-seq and ChIP-seq to study this mutation and this p53 mutant as a transcription factor. And we also use more target approaches such as immunoprecipitation and subcellular fractionation methods to study the interaction of p53 with other proteins. So we found that A347D is hyperstable and yields a mutant p53 protein that preferentially forms dimers. And despite this hyperstability, we found that this mutant has completely lost canonical p53 transcriptional activity, which is a loss of function that is likely tumor promoting. On the other hand, we found that this mutant p53 induces mitochondrial network changes and is able to directly associate with mitochondrial proteins to induce apoptosis in a transcriptionally independent manner. Additionally, we found that cells with this mutant are hypersensitive to topoisomerase II inhibition, which may be exploited therapeutically. And this really suggests that this mutant p53 exhibits both loss of function and gain of function properties that give it almost a double-edged sword property, in which loss of function is tumor promoting, whereas the gain of function activity is almost paradoxically tumor suppressing. So to our knowledge, we're the first to demonstrate that a mutant p53 is able to induce cell death through direct mitochondrial apoptosis after topoisomerase II inhibition. And although this is at a very preliminary stage using preclinical models, our work really suggests that topoisomerase II inhibitors might be more effective for Li-Fraumeni syndrome patients with this mutation. And additionally, this study sets the stage to assess whether other more commonly occurring p53 variants may also exhibit this pro-apoptotic ability that may be exploited therapeutically. So as I mentioned previously, we're interested in how other hotspot mutants might be differentially sensitive to certain chemotherapeutic agents and may exhibit properties like A347D to directly induce mitochondrial apoptosis. Thus, we're examining other hotspot mutants that are associated with Li-Fraumeni syndrome to see whether they also retain the license to kill. I'd also like to mention that this work was performed primarily in the lab of Carol Privis at Columbia University and was just published last week in the Journal of Cancer Discovery. So if you'd like to learn more, please check our paper out.